I said this is like a family, right? There is like a
Yeah.
grandfather and a father, uncle, brother. Each
Grandson.
stream there is no sister, right? Is there sister over there?
Oh, this is Nick.
Hey, Nick.
Hello.
Oh, hi, Nick. There's too much
Someone's
You
on the
seem
phone
to have
and
reverberation.
they need to mute their microphone.
Who's causing? Is it me causing this reverberation?
Someone else.
Okay.
No, I think Ken and I are in the same conference room, so there is some
You.
So, Arun, you're going to have to sort that out or we won't be able to hear you guys. Can you say something again, Arun? Arun. Can
Yeah.
you hear?
So
Arun,
if I turn it on, then there's a lot of background
one
noise.
of you needs to mute.
Yeah, so I have to.
Now, Ken, can you talk? Ken, can you hear us?
Hey,
Yeah.
I'm
Sorry,
simply
we won't
unmuted.
hear you guys.
Should I try one?
Hey, Doug, how are you doing? You're on mute, Doug.
Where's Doug?
He's in the bottom right-hand corner.
I am here.
Okay, why do I only see half of the next second row of people? I should be able to see everybody. Oh, there's Doug.
I knew the first
Hey,
session
Doug.
would be just trying to get on the computer.
Hey, Nick, how are you?
I guess this is not the old Tandy computers.
You mean the one that ate my thesis?
Yeah, where's my floppy disk?
So Arun, were you able to fix that? I can't hear you, Arun. You're on mute. Mute. Arun, you're on mute.
Is it better now? Can you hear me now? Yeah.
We keep kind of hear you. Ken, can you speak? Can you go off mute and speak? No, you're on mute, Ken.
Okay, I'm unmuted now.
Yeah, okay, we can hear you. And Dirk just joined us. Hi, Dirk. How are you doing?
Hi, Jude. Hi, everyone. Doing good. How's everyone else?
Oh, we're getting there. It's not the Grinch that stole Christmas. It's the Grinch that stole that stole our computer. So, is this everybody? Can I assume this is everybody? Okay, I'm gonna make some rules up just to ensure that we hear each other and facilitate. One is, if you're not speaking, be on mute so that we don't get reverberation. And we could try this, which I don't think it's going to work, which is, raise your hand. And then they know we know you want to ask a question. And then if you hit it again, you can lower your hand. But if that doesn't work, which I know it's not going to work, we can just stick with whatever we're doing. But the way I thought we would initiate this was fairly simple. We'll go around, everybody knows everybody, but I'm recording this. So I'm actually recording this because there's intention at some point where we either want to transcribe this and publish it, or we'll just keep it so people can get access to it over time, because there's such a wealth of information sitting on this Zoom. It's not fair to the rest of the community what you guys have and continue to hold as far as information, especially stuff that never got published. And I think out of respect for at Chengwen, there's the grandfather, the father, the son, the grandson are probably on these screens. I think Ken needs to give us a few initiating words on how all of this got started. And then I was going to show some slides to just point us in a direction for some topics to discuss about. We won't get through them today, but I thought if we at least got exposed to them, that everybody would begin to harmonize on what are real significant issues that are going on in the field today and things that were left undone. At the same time, there's just too many topics to go into and too many publications that we want to ask questions about. And I would just say, for the sake of the next generation, it would be extremely valuable if there was a historical perspective to your response, as far as when did you uncover something or see something or study something that you may or may not have gotten an answer to yet. So with that kind of brief intro, I was going to ask Ken, if you would just start us off with a generic, how did we all end up where we're at today? And I think it came from some of the early postdoctoral studies at the NIH with you guys, and being curious about a virus that didn't seem to be pathogenic.
Okay. I was defending the country at the NIH in 1968, and I was working on vaccinia. But I was approached one day by Jim Rose, who was working on AAV. And Jim was very agitated because Lionel Crawford had published a paper saying that AAV was a single-stranded DNA virus, and Jim had just published a paper in which he said the DNA was a linear double-stranded molecule that was twice as big as it should have been. So he wanted to know, could we resolve that problem? And I said, I'll come back to you tomorrow and tell you. So I came back the next day and suggested a takeoff on the measles install experiment. And it was very simple. We made one preparation that was high density DNA and one that was normal. And if we mixed the viruses together before we extracted the DNA, we got out DNA that was of hybrid density. If we extracted the DNA first and then mixed the two preparations, we got out heavy and light DNA. Therefore, it was pretty clear that the DNA in the virus was single-stranded, and that it hybridized to the double-stranded form after purification or during purification. So that got us started, and there are a variety of studies, and I went to Hopkins about a year or two later on the faculty, and while there, I picked up one postdoc named Bill Houseworth, and so Bill he wanted to learn molecular biology, he claimed, although he was already a faculty member. And so he worked with me. We worked on replication of the virus, and we did a lot of other things like physical maps. The one thing that we discovered, of course, during that time was that AAV DNA had a palindromic terminal repeat. The only problem, the first problem was that the terminal 5' nucleotide was variable, and we couldn't figure that out till we realized that the 5' terminus read TTGG, TGG, or just GG. And so, as we learned later, that was a reflection of some imprecision probably in the nicking during the replication process. Anyway, a few years later, I moved to Florida as the chair, and Houseworth decided he wanted to come with me, which he did. Some guy named Muzycka also agreed to join us. He at that time was a postdoc with Dan Nathans at Hopkins. And then Jude, somehow you became a graduate student. I don't know if I should tell how you became a graduate student, but it was pretty amazing. I guess in this group I can tell the story. Anyway, Jude was at Yale working with Charles Radding. And actually, he was running Charles's lab and designing many experiments on his own, writing his own papers, etc. But he was a technician, and he had a less than stellar undergraduate record at Clemson, as I remember. Anyway, one of our new faculty was a guy named Bill Holloman, who came from Yale, and actually, you were working with Sherm Weissman. It was Bill that was Radding. Anyway, so Jude, he said we should recruit Jude as a graduate student. And that was a little bit of a challenge because the assistant dean of the graduate school was a difficult personality. Anyway, we managed to get Jude over the hoops. We did have to let him take the test of English as a foreign language, which he barely managed to pass, and the rest is history. By the time he graduated, he was giving the best talks in the department, I think both among the faculty and the students. And he went off to Princeton with. Did you go with Tom as a postdoc? Yeah, and so that was that, and I would say that Jude played a major role in getting the first AAV, or one of the first AAV patents, which included Nick and Paul Hermanot, and somehow I got put on the thing as a gift, I imagine. And but it was Jude who figured out how we get through the patent office and make the thing work, and it did work. And for a couple of years, that patent was second only to Gatorade as a fundraiser for the University of Florida. It wasn't quite in the same league, but it was not a bad patent. And if the people had paid us what they should have paid us, probably would have brought in even more money. Anyway, I think that's the history. Other people came through. Some guy named Srivastava became a postdoc in the lab, and he bailed out while I was out of the country, and it took us 2 years to rescue him from where he went to Arkansas and get him into a reasonable place in Indiana, where he really began to function well. And eventually he came back, I left and eventually came back, and Nick left and eventually came back to Florida. And and some guy named Doug was a graduate student here. I think, were you with Nick or were you with Bill?
I was with Bill then.
Yeah.
And then went to Stony Brook with Nick.
Yeah, that's what I sort of remembered. So that's a brief history from the Gainesville area. Can't hear you, Jude.
Jürgen, you have to speak a word or two about you and Dirk and the Zur housing contingency that got started over there. And then we can move into some topics that I've prepared for.
Sorry,
Go ahead and tell us your history.
my history.
Yeah.
I actually started to work on chromatin assembly in Heidelberg, working with Xenopus, and we were in competition with a group in Cambridge who actually first named a chaperone molecule, so nucleoplasmin, and we were able to isolate the first chaperone complex of a protein with bound with two histones, histone H3 and H4. And we could then show how nucleoplasmin and protein N1 together assemble nucleosomes. So this was my starting point. And at that time, I was also involved in the regulation of actin in the nucleus, where we could first show a regulator of actin polymerization located in the nucleus. And I also was a bit involved in proteasome detection. But all this work had been done with Xenopus laevis oocytes, and I was at the Cancer Research Center. And I didn't feel well to work with Xenopus oocytes in the Cancer Research Center. And I tried to find a subject which is more related to cancer research. And at that time, Harald Zurhausen started to work at the Cancer Center, and he brought in the project with AAV. Actually, they discovered first the AAV5, I think. And at that time, it was hypothesized that AAV2 is able to inhibit cancer, is able to, especially it has an inhibitory function against human papillomavirus infections. And this was the starting point that I started to work with AAV. I changed completely the subjects, and I remember that in the year where I moved to the AAV field, I did read in my vacations a description of AAV and everything from Ken Berns. And this was for me the Bible to understand everything. And it showed me how to enter and what to do. And the first work was we wanted to analyze the influence of AAV and the Rep proteins on the transcription of papillomavirus genomes. And but after a while, I recognized that AAV does not really inhibit papillomavirus transcription. And then I changed to the other subjects, which you probably all know what we have tried and so on.
Awesome. Thanks, Jürgen. Dirk, do you want to spend a second just introducing your relationship and what you're doing now?
Sure. I guess I joined your lab, Jürgen, at the time when you just underwent this transition phase that you just mentioned from papillomaviruses to the other subjects, which I guess includes vector production, because I joined your lab in 1995, and I, of course, met one of your PhDs, Markus Hörer, who was working on the project you just mentioned, a close friend of mine still until today. I've also switched topics completely, like Jürgen just said. I also started out at the DKFZ as a PhD, not a PhD student, as a diploma student, the equivalent of a master's student today. Worked on a completely different subject before I came to Jürgen's lab. Very quickly, I worked on casein kinase 2, CK2, and I think in retrospect, it was a lucky development that it turned out. The reason why nothing worked in my roughly 1 year that I worked as a master's student was that it's more or less funny, but in retrospect, it turned out that postdoc, she had faked all the data that my work was based on. So my project was to basically unravel the mechanism how one subunit regulates the other, and I could not. I spent like literally 365 days in most of them in the hot lab, running sequencing shells, northern plots of all kinds, and nothing ever worked. In the end, I quit the group because it just didn't work. I finished my master degree, but just nothing worked. And then after I joined Jürgen's group, it turned out the postdoc was fired shortly after, because it turned out she literally used pure protein to paint Western blots.
Okay.
So it was all bogus. So that was my start in science. Not a good start. That's why I also desperately looked for a new topic. And at the time, Jürgen had already worked on AAV. So that was, after one interview, was clear this is absolutely what excites me, what I want to do. And then I joined his lab. And then, yeah, as Jürgen said, from there on, I guess you all know what we've been doing, what I've been doing in Jürgen's lab. Like you did, Jude, of course, the one thing that I focused on as a PhD student was to develop these helper-free production systems, which we all both published at the same time, more or less. And then from there, I stayed in Jürgen's lab after my PhD for 2 more years. And then I guess most of you also know this, went to Mark's lab and then came back to Heidelberg in 2007 and started my own group here. We still work a lot on vector technologies, but also, of course, on library screens, high throughput screens in vivo, most lately. That's
awesome. Thank you.
Rundown.
There's 3 other people on the screen that some of you may know, some of you may not know. Down in the center is, bottom is, that's Matt Hirsch. Matt Hirsch is a faculty member at UNC Gene Therapy Center. He came into the lab as a postdoc and then independently generated his own topics and that. And Matt at the moment's working on putting AAV in the lens of kids who have MPS I disorders and they have cloudy corneas, and he's been able to show that he can clear the cornea by putting the enzyme back in there. It was on the cover of Molecular Therapy. Matt's been working on some fundamental things back to what's the role of adenovirus and AAV, and how does AAV infectivity and stuff like that. The bottom left, this is like Hollywood Squares, is Weidong on my screen. Weidong was the second student to come into the lab when we were at Pittsburgh. And Weidong came in with a physical background. He knew nothing about molecular biology. And the first week in the lab, I think 3 members came in and said, you need to make this guy leave. He doesn't know how to do anything. And I think Weidong spun a tube of radioactivity and it went all over the lab. And so everybody wanted him out, and we persisted. And Weidong now is an independent faculty member at Indiana University with Roland Herzog, and has independently generated some of the early studies on HIV expressing rep and things like that. Too many to discuss. But Weidong has a study, Ken, that I think is relevant to what you were talking about, the terminal repeats, and we'll hopefully get to talk about some of that today. And then in the top left-hand corner on my side is Chengwen Li. Chengwen Li is another postdoc that came into the lab a number of years ago and has independently built his own career. And besides that, he has played the role in managing most of the gene therapy center when I've been too busy and stuff like that. But Chengwen has uncovered a number of the immunological concerns with the AAV vectors, and has pointed to some really intriguing observations that I think have gotten lost in the community. Basically, that the ITRs have promoter activity that Terry Flott described a long time ago, and that Chengwen's been able to show you can make double-stranded RNA because of that, and you can trigger these innate immune responses, and it may be one of the precipitating events that causes toxicities in humans. And so I invited them to sit and primarily listen and offer input when we end up getting stuck on something that we don't know. But I wanted to take at least 10 or 15 minutes to show you some slides. And I'm doing this primarily to set up a conversation. I think it's already relevant what Ken mentioned about the early studies of AAV and the role of the ITRs and the putative nicking at TTG versus TG versus G. I have a teaser in here to just have you think about it for a second. But more important, I was hoping we could focus back on data that was either not published or published and not understood, or published and not only not understood, but still outstanding as to what to do next. And I know Doug has gone down a career of looking at AAV as a potential insertional mutagen and the role that it's played in jumping into the chromosome. And from some of his studies, he gave the impression it's incomplete genomes or DI particles that Barry Carter described that are the primary culprits. And Weidong just published a paper showing that there seems to be a mechanism where these things are being generated, not only in wild type, but in the vectors. And so there's some biology here that's going to plague the field. But bear with me for a second. Let me set the stage if you guys don't mind. And if you can see my screen. Can you see this okay? I'm assuming.
Yeah, you're up, Jude.
Okay. So again, I wanted to start this. This is the classic hula, what are we doing here kind of thing. This is the AAV virus, thanks to Mavis primarily for all of the information we know at the structural level. But there are a ton of papers that have come out by multiple groups. Jürgen and Nick is the ones I pulled because there were some of these things that ended up having outcomes that didn't make sense that I think are impacting some of the data that we're seeing today in the clinic. And I just threw these up here just so people would get a sense of all the things that are still out there that need to be pursued a little bit more. This was done by Chengwen Li, and the reason I showed this is because I think we started to gravitate towards a common mechanism of how AAV infects the cell, binding to its primary receptor, clathrin-coated pits, endocytosis, early endosomes escaping, eventually either being ubiquitinated, where Arun's made a significant contribution, arguing that if you block these steps, you can prevent the virus from being digested in proteasomes. Some mechanism of entry, and then a step here, which I think is really fascinating, this so-called uncoding step. And I want to focus on this because I think this is a primary driver in what's happening in the clinic today, and we don't clearly understand it. After that, we know the story of whether it goes through second strand synthesis in order to be a functional template for transcription, and then making RNA to actually make a product. So while this is supported by many, many studies from many, many people, I don't have the time to go through who did what. It's just kind of a backdrop to think about when we look at some of the data that's coming up. This is an ongoing clinical trial that's now treated 30-something kids. There's 100 and I think 19 of them in the world. It was done by Chris Bankowitz. He's using just AAV2, and he's putting in the gene amino acid decarboxylase, and he's injecting it directly into a structure that if it's successful, he's able to make dopamine, and the dopamine rescues back the function that's missing. This is typically referred to as juvenile Parkinson's disease because these kids are frozen. And everything about this is similar to the Leber's congenital amaurosis, where all of the structural integrity is there, but it's just missing an enzyme. So it's not building back circuitry or anything. It's simply putting back an enzyme that's missing. And I just show you this because these are these kids. They can't move their body parts. They don't sleep. They have seizures. They have to be hand-fed most of the time, things of that nature. And at the same time, for the parents, it's an incredible, horrible disease to have to try to manage. After Chris does a single injection in these patients, what you end up seeing is a transformative event, but it all happens around the same time at 18 months. You can see her over here beginning to dance to the music. You see the brother will egg her on at one point when he teases her with his shoulder, and then she starts making additional movements. And she responds back. And then as time goes by, every one of these kids is improving to the point where their ability to communicate, function, participate is just overwhelming. It's completely overwhelming in the sense that Chris has now rescued like 30-something kids. He's in Poland now treating 2 kids, one from Israel, one from the Netherlands, and he continues to do this. And at the same time, he just published this study. We get the impression that you don't need a special vector, you don't need a special capsid. You just need to be able to get it into the target area. In this case, it was the midbrain. Chris made a comment which I thought was interesting. He said, no matter if the kids were 1 year old or 10 years old or 18 years old, the cascade of events took place exactly the same in every one of them, suggesting that when the virus infected, when it migrated to the nucleus, when it uncoded, when it expressed the genes, it was on a case, it was on an exact same path in every one of these patients, regardless of the age or development, and he had the same outcome of biological effect, suggesting that the gene expression was behaving identical regardless of what the patient's development of disease was. So plant that aside and think about it for a second, because at least in a very human biological system, there's a program of what happens almost to the T with respect to a clinical outcome, which would argue very specifically to a molecular outcome, which goes back to a viral infectivity that we just looked at a second ago. Now I'm going to show you this piece of data because this was generated and it's completely puzzle to us at the moment. And it's a simple injection of an AAV virus called AAV2 retro, which is supposed to be a virus that after you inject it, it will retrograde to other parts of the brain and express the gene product. In this case, the colleagues who developed this vector used a human synapsin promoter, which is a neurological promoter for neurons. And they demonstrated this, and we were able to take an identical cassette and put it in monkeys and show that indeed, if you went through and looked at the other parts of the brain, you could see expression. But then the colleague actually took the same vector trying to get a stronger expression and put in the CGA promoter. And of course, we all know this is a very strong, ubiquitous promoter. And when he did this, he had tremendous expression at the site of injection. But when he went to see where the vector was in the retrograde, there was no evidence for expression. And we were totally puzzled because both of these are the same capsid, which would argue that they should get to the same location. And yet, by simply changing the promoter, we had one result that was negative for expression in a distant target cell and another result that was positive. And this puzzle has been out there for a while and just, I think, reproduced by various groups over and over. And for me, it pointed to a paper that Nick ended up putting out, which suggested that there were mutations in the two-fold interface that were responsible for suggesting a role in viral transcription. Now, this is very puzzling because the obvious answer would be, oh, it's an uncoding mutant. It won't let go of the DNA. And I think Nick carried out all the experiments to suggest that it's not uncoding, the DNA is free and so forth. And yet you don't see transcription taking place in this virus. And I bring that up as there's a mutant that seems to suggest there's a role for the capsids in deriving viral transcription. There's biology that says the same capsid with different promoters are giving completely different biological outcomes. And yet, in this clinical trial with this specific cassette, everything worked exactly the same. And so I put this out there because I think what we haven't figured out yet is what is happening at this level of gene expression. And I want to paint this theme because Arun turned me on to a problem that I think we saw and have seen for a number of years, and that was the amount of multiplicity that it takes to transduce a cell. And again, I'll show you a simple example if we consider the brain, the petri dish, there's 100 billion cells there. The liver, there's 240 billion. If you do muscle, there's around 20 trillion. So total, there's about 30 trillion cells. So if you're going to add AAV, IV, and you want to get to this target cell, you would expect a multiplicity of 240 billion if it was 1, and 10 if it was 101, or whatever the multiplicity would go out. But Arun actually tableized this data, which is all derived from clinical trials that have been done to date using numerous AAV serotypes at varying doses, and the clinical outcome was marginal at best for all of these. And yet the dose that was being used in these studies, in the case of BioMarin, it's 147 times the total number of cells in the body. And so we're putting in these enormous amounts of virus and getting low levels of transduction that's suggesting that the capsids are not getting to the right target or they're not expressing. And this launched about 10 years of capsid libraries and things of that nature to try to sort this problem out. But I don't think the capsids are the issue, and I'll show you some data of that in a second. I wanted to show you the last piece before we start talking, which was Xiao's work of putting a mini dystrophin gene into a dog model where they typically die at 6 months, and that he was able to completely rescue these dogs, and not only show that he rescued them, that the dogs actually did really well for the life of the animal. The animal was put down around 8 or 9 years because of some heart problems, and we thought that was due to the CMV promoter breaking down over time. But anyway, it was Xiao's data with this mini dystrophin that led to a clinical trial that was done at Duke, and here's the first patient, his name is Connor, and Connor was treated by Eddie Smith, who was a clinician, and when he gave him the virus, again, it was trillions of billions of viruses done IV while he played with his computer game. And in order to get in this trial, they had to walk up steps, 4 steps before they could enter. And if you look at the left, this is 5 days before the procedure. You can see Connor and him going up the steps. And then you can see basically a month later, his ability to walk. This is his electric chair that used to take him upstairs. He doesn't use it anymore. And yet with all of these studies, this data was so compelling in these ambulatory young kids that Pfizer's taken this to a phase 3 clinical trial. We heard today that a patient died today from this treatment, and we're completely perplexed as to what happened, because there's been no concerns of this throughout. They're using the mini dystrophin gene that Xiao generated. They're using the same, procedure that we generated and putting it into their patients. And yet they went for the first time into a non-ambulatory patient. And we think that it generated a toxicity that probably pushed them over the edge and caused the death. And we now know that Adontis has had 3 kids that have died because of what's referred to as high-dose AAV toxicity. This is the first one in DMD, and I'm afraid it won't be the last. And yet, if we go back to this biology, we're dealing with AAV particles that get into the cell and don't appear to be functional. And I want to share this data with you that was first generated by Jim Wilson and independently by Livio Danos, where he took a comparison of looking at mouse versus monkey using different doses of AAV and he was scoring a transgene. And in this case, you could see there's a dose escalation that takes place from 10 to the 12 to 10 to the 14. But this is on a log scale, and you can see he's, almost at 1.5 grams of protein being made. And the same dose put into a monkey, you can see it's almost a log off in its ability of the amount of material being generated. And so again, everybody continues to look at this. If the capsid's gotten into the mouse, maybe it won't work in the monkey. And we think the same is true for humans. And yet when Olivier did this experiment where he looked at DAPI to show if the liver cells had AAV in it, he could show in the mouse and in the monkey, there was no difference in the amount of virus transducing these two organs. But when he looked at expression, the mouse was expressing like crazy, and the monkey was rarely expressing anything, showing this complete dichotomy between two different species, not at the DNA level, but at the RNA level. And I bring you back to that study where we were talking about AAV in the brain with two different promoters, when one it was expressed and the other one it wasn't. They went on to quantitate this, looking at genome copies per microgram. Again, you can see the mouse versus monkey, they're near identical at all the doses, but at the expression level, they're completely inconsistent with the amount of genomes that are present. And then they did one more really clever experiment where they took a mouse model where you can put human liver inside in the mouse and make a humanized liver. And I think both Weidong and Dirk and Chengwen have had success with this. And using this model, they were able to show that 70% of the liver was basically humanized. And they used different staining techniques to see the mouse versus the human, the vector DNA versus the RNA. And once again, the data screamed at us that the human livers were not expressing, whereas the mouse livers were. And for me, this is a major issue that has continued to pop up, where the clinical community has answered this by saying, put in a higher dose, put in a higher dose. The patient that succumbed today had 3 times 10 to the 14 particles per kilo, an extraordinary amount of virus inside of the individual. And at this juncture, we don't have a sense as to why this silencing or inability to express is taking place. And so I bring this back to the group with no answer, but simply pointing out that there's some molecular biology experiments with capsid structure by Nick and the group saying they can find specific amino acids that seem to be involved in the mechanism not yet understood about whether you transcribe or not transcribe. There seems to be a species difference that takes place that Chengwen showed, that if you go from mouse to monkey with the same vector dose, you'll get 200% factor 9 in the mouse and less than 40% in the monkey, and then in the human it's 2%. And yet this barrier, I think, comes back to what I think Ken started the whole talk out with, which is the AAV terminal repeats. It's the only thing that's common in every one of these experiments, irrespective of the capsid, irrespective of the transgene, irrespective of the disease, irrespective of the production, is a inability of these genomes to be functional when you go into higher primates. And so I was going to stop and ask Nick to at least start us off with whatever your understanding of the so-called twofold mutants that were blocking transcription and what you think might be at play, or more important, what experiments did you guys start with to suggest this was the only alternative left for why these mutants were dead mutants? And you have to unmute first, Nick, so that we can hear you.
Hear me?
Yes.
Okay. So this was a problem that I retired during this problem. And I retired because I decided that it was too complicated for me. It started when Mavis did a really clever experiment. She took her crystals of AAV2 and she did the experiment again with AAV, I believe AAV8 and AAV5. And she soaked her crystals in pH, and she first determined the crystal structure at neutral pH and then soaked them again at pH 6 and pH 5, I believe, if I remember correctly, and redetermined the crystal structure and found two regions that where the amino acid actually shifted. So these were pH dependent shifts in the structure of the capsid. And the first region that shifted was a set of amino acids at the twofold interface. They were on the surface. They were to some extent involved in maintaining the integrity of the capsid, but really they did not serve a major function in holding the capsid together. They were out there on the surface interacting with the environment. And the second region that changed with pH was, for some reason, I now have slides in my screen. But anyway, the second reason that changed was internal in the capsid. Oh, thank you very much. So this is the so-called dead zone. So this is the surface. This is the twofold interface. And what you see in this picture is a series of amino acids. And each one of these amino acids, when you mutate them by themselves, have the phenotype that they are absolutely completely dead. And when I say dead, I mean you cannot detect any infectivity, any expression, any gene expression whatsoever. So they are the limits of our detections were approximately 6 logs lower than the wild type capsid. So, and this is unusual because many of you have made mutants. Mutants tend to be down 2 logs, 3 logs, 4 logs. Those are very tight mutants. You don't see mutants where you absolutely don't detect anything. And we were not detecting anything with these mutants. And they were all in this one region, which had previously been identified by the group at Avogen and been called the dead zone. They had done a series of surface mutations, and mutations in this region tended to be much lower in infectivity. So we asked the question, I mean, we immediately assumed these were receptor binding sites. That would be the simplest explanation. But it turns out these mutants got into the cytoplasm just fine, as efficiently in most cases as did the wild type. And so then we said, fine, so they're not getting out of the endosome. Well, it turns out they were getting out of the endosome. Okay, fine, then they're not getting into the nucleus. Well, it turns out there were changes in the rate at which they got into the nucleus in some cases. But at the end of the day, at 48 hours, they were all getting into the nucleus at about the same rate. Roughly 50% of everything that got inside the cell wound up in the nucleus as capsids. And then we said, okay, so we have an uncoding mutant. This is interesting. We haven't seen one of these before. So we isolated the virus from the nucleus, and we asked, okay, how much of it has now extruded their DNA? So that would be then the DNA would then be DNA sensitive in the nucleus. And then let's ask what fraction of it is still virus where the DNA is still protected from DNAs. And the answer was, they all uncoded. There was no problem in uncoding at all. And so finally, we had to consider the possibility that this was an expression problem, and we searched for RNA. And we saw absolutely no, or almost no, messenger RNA from these mutants. So we had one other tool, and that was Jürgen's A20 antibody. This is a very unusual antibody because it recognizes, as Jürgen has shown, it recognizes only intact, fully formed capsids. And when we isolated these capsids from the nucleus, as Jürgen showed, the A20 antibody was there along with the capsids. So and Jürgen showed this a while ago. You could bind the A20 antibody to the capsid, but the capsid would still enter the cytoplasm, still get into the nucleus, and still and be in the nucleus. And I believe he even showed it uncoated. Okay. So we said, okay, maybe the antibody will do exactly the same thing as the mutants are doing. And so indeed, we did the experiment. We bound it to A20 and we infected cells, and the phenotype was identical. No transcription. It got into the nucleus, it uncoated the DNA, but it absolutely killed transcription. So A20 antibody kills transcription, period. That's the mechanism of the A20 antibody. So what is it doing? And how is it uncoding and yet somehow still having an effect on transcription? You begin thinking about possibilities like, well, yes, of course, the DNA is extruded, but the capsid holds onto it through the terminal repeat. So it's outside the capsid, but it's still attached to the capsid. And the capsid is sitting very close to the promoter region, and somehow is interacting with cellular factors that are either this capsid is either recruiting a very powerful repressor function, okay, or it's recruiting, it's failing to recruit a very powerful transactivator, okay. And that's where we stopped the problem, and I closed the laboratory, and hoping one of you folks will take over and solve the problem. But it has to be something like that. It has to be, and in order to solve this problem in a molecular biology or biochemical way, you almost have to begin looking at what things are bound to the promoter regions after this, after encoding in the mutant versus wild type in the nucleus, or you have to look at what other proteins, what complexes, what's in the complexes. You know pretty well particularly with the AAV promoters, the wild type AAV promoters, you know what's bound to the DNA at the P5 promoter region, for example. And you can imagine then what kinds of things that would recruit. And I'm guessing that we're recruiting the entire mediator function of some 30 different proteins through YY1 and REP. I don't know what we're recruiting through the CMV promoters that many of us are using, or through something like that's neural specific, like the alpha-synuclein. But presumably these housekeeping transcriptional factors, mediator and these other complex factors, are involved in promoting transcription from the AAV vector, and the capsid is somehow determining whether that actually is allowed to happen or not happen. And that may well be cell type specific. It may happen in some neurons, but may not happen in liver, for example. So these are the kinds of things that we began thinking about. And in fact, we were at my last grant proposal, these are the kinds of things we began proposing. We did propose to study, and then it became time for me to quit while I was ahead. And so that closed the lab. But I hope you guys will take over on this, because it's a fascinating problem. And there is no other example that I know of where the structural proteins of the capsid are actually transcription factors. That's bizarre. That's not supposed to happen. This is just a delivery vehicle. It's just supposed to drop the payload in the nucleus and go away. And it's not doing that.
So I have a question primarily towards Jürgen and Dirk, which feeds off of that, Nick. And that is, when you talk about packaging, you know the ITR is essential, and the different mechanisms argue that REP is threading it into the capsid protein, and the virus is holding on to it. How does the virus know when to let go of the DNA? Because like you said, if it lets go in the cytoplasm or outside of the cell, it's a suicide mission. If it does it at the right place and the right time in the nucleus, it's able to go forward.
So let me tell you about the other set of mutants that were pH sensitive in Mavis's crystallographic study. There's a set of 4 amino acids. This is going to go on for a few seconds. I'm sorry.
Call from 406303589.
There's a set of 4 amino acids at the threefold interface where if you now think about being inside the capsid and you look at the threefold interface where the 3 molecules come together, 3 proteins come together, there's a set of 4 amino acids in 1, 2, 3. So there's a threefold of these quartets. When Mavis does crystallography on MVM or AAV, she finds an adenine residue bound to that internal threefold, and that this work is not in this paper, Jude. So you don't have to put up the slides. This is stuff that hasn't been published, and it's my fault that it hasn't been published. Anyway, so obviously we wanted to mutagenize these two to find out what's going on. The interesting thing about this is that when she does the low pH, pH 6 and then pH 5, that adenine residue, so what she found from way back from MVM crystallography was that at this internal threefold position, there is an adenine bound to this quartet of amino acids. They, by the way, are phenylalanines. So it's believed that we believe that the adenine is held by what do you call it, van der Waals forces, by base stacking, basically. You drop the pH, and that adenine signal disappears. It's no longer there in a crystal structure. So we said, okay, so this makes sense. You drop the pH in the early endosome. You're now releasing the DNA and allowing and preparing it for extrusion out of the capsid. Okay, that's great. This is going to be wonderful. So we make the mutants. We make them one at a time, and we see hardly any effect whatsoever. We make perfectly good virus. It's still infectious, blah, blah, blah. And we're going, oh, okay, that was a big nothing. And it's now on the back burner, and I tell one of my technicians, well, we should do these mutants 2 at a time. Let's make them just pairs, 2 pairs, 2, and then do 3, and then let's mutagenize all 4 of them and see what happens. Maybe it's a cooperative effect. When we start doing 2 at a time, all of a sudden, we can no longer assemble capsids. This is not an infectivity problem. This is now an assembly problem. We can't make intact capsids anymore. There's one mutant that's particularly important. That mutant in combination with any one of the others kills capsid assembly. So this doesn't make sense either, because Jürgen's model, and I believe in Jürgen's model, is that you have an intact capsid that you probably use the intact rep 78 or 68 that's still covalently bound to the DNA to attach to the capsid. Okay, that's how you tell the capsid, oh, this is an authentic AAV molecule because it's got a rep bound to it. And then you bring the 3' hydroxyl group, the 3' end over, and rep 52, rep 48 starts threading the thing into the capsid. And then somehow magically at the very end, you precisely get rid of that rep, the covalently bound rep. I don't know. That's the other mystery step that nobody has ever really figured out how that happens. That's fine. But so in none of this model is there provision for there being a problem with assembly of that because of mutations at the interior threefold axis mutants. Okay. They should not be affecting assembly of empty capsids, and yet they are. Now, the other thing that Mavis saw years ago and is repeatedly seen is that even when you make empties, so if you take the adenoviruses that have the capsid genes in them, and so you express them, and so you make tons of empty capsids, there's no AAV DNA, no TRs, nowhere in sight. And you put those caps and you isolate those capsids and do crystallography on them, there's DNA inside. And the DNA is at those threefold positions on the internal side. Sure enough, with adenines attached. Okay, adenine seems to be the molecule that's used. So where does that come from? And why is it there? And why do those mutants have an assembly problem? What that's saying to both to us, what it was saying to us, is that we might be using DNA to some extent as a scaffold in order to create an intact capsid. That may be how you assemble the capsid to begin with. And for that purpose, you may be able to use any piece of random DNA that's inside the cell. Then you have the other problem. If that so readily happens, then while you're threading the AAV DNA in, how do you get rid of that stuff? And because when we do, in fact, we've never really done this experiment. We've never taken a highly purified AAV virus stock, isolated the DNA from it, maybe Ken knows, put it on a gel and asked, are there small molecules in there? Because the small molecules in so-called empties are about 200 bases long.
200.
Yeah, give or take. They're a smear, but they're averaging around there.
So, Nick, let me just add to that storyline. If you purify AAV particles, and we have an ongoing trial with empty particles being put into healthy volunteers to see what the toxicity profile is, you get the same amount of DNA contamination in those empty particles as if you were making AAV with a vector genome inside of the particle. So it's similar to what you just described. There's DNA affiliated with the particle, irrespective of if you make it empty or with AAV vectors.
Well, it can't be the same amount because they go in a light position in the cesium gradient, right? So it's got to be less DNA total.
It's less DNA, but I'm saying there's DNA associated with it. And the FDA has screened that as over and over because in the monoclonal antibody world, they can reduce the amount of contaminating DNA down to like a picogram per milligram of protein. And we're still stuck with this like nanogram to microgram amounts of contaminant. No matter how much you DNA it or go put it through all of these machinations, it's either so tightly associated with the virus or it's inside of the virus that you can't degrade it and get rid of it. I'm going to ask Jürgen to comment on the empty particle being the substrate for packaging. You're on mute, Jürgen.
I think this can be expected if you have an efficient assembly system that it includes small molecules during the assembly process. And I think this is just the normal physical happening. What I want to say for assembly of or packaging of AAV, it is always discussed as if capsids are formed and then wrapped together with the genome binds to the capsid. But this is definitively not the case. If capsids are assembled, RAP is not able to bind to the capsid. So the association with the capsid occurs during capsid assembly. So a RAP must be present with the capsid proteins during the assembly process. And there is some initiation or some interaction which allows then to pump or somehow bring the genome into the capsid. So it's not assembly of ready capsids and then association with the proteins and the genome. And in the early paper of Myers and Carter, where they established the basic idea of the packaging process. They describe AAV SP product, which is a product with high density, so it must be DNA associated with the protein, but it has a low sedimentation coefficient, which means that it has an open conformation, and this could be the intermediate or the packaging initiation complex, where DNA is associated with capsids, but in an open conformation.
Yikes. This changes some of the thought process on packaging.
Let me ask a question from Nick, though. Can you put a model like that into the context of the in vitro packaging that you had done at Stony Brook with Chengwen? Because that was done with pre-assembled purified capsid, I think.
Nick, that question's for you.
I'm sorry, I didn't catch what Doug was saying.
Doug's asking if you take your in vitro packaging system, it was our understanding that you had assembled capsids that you were putting the replication complex together with and able to package, and not a just AAV VP123 that were disassociated. Is that right, Doug?
Yeah, well, you know, let me comment on that. So we became convinced at the end of the day that replication was a part and parcel of packaging. If we did not have rep protein, we did not package. And we're talking about the large rep proteins, because we used your rep 7868 antibody that's specific for just the large rep proteins to do, for example, to deplete our extracts, and that killed packaging. So, and then we also did the classic acetylcholine experiment. As soon as we added acetylcholine and stopped replication, we knew by many previous experiments that it stopped replication completely. Then again, there was no packaging. So it was clear that we needed ongoing replication while we were packaging. And the extract that we prepared to do that was in fact a replication extract. It was a classic Tom Kelly, Bruce Stillman, 0.2 molar sodium chloride extraction of cells. It was essentially a cytoplasmic extract with a 0.2 molar sodium chloride extract of nuclei. That doesn't get everything out of the nucleus. It gets what you need for DNA replication, and that's what we were using for isolating the cellular proteins for AAV DNA replication. And we knew we would have capsid proteins as well, and presumably would have AAP protein, although we didn't know about AAP protein at the time. So there's no question we needed replication simultaneously with packaging. Otherwise, we killed packaging absolutely cold. There's nothing going on anymore. The other thing about that in vitro system is that we got two kinds of particles, and they were present at the end in about equal amounts. One of them was, by our criteria, we used the criteria for an authentic capsid, which is it had to be chloroform resistant, it had to be heat resistant, 55 degrees for I think 30 minutes, and it had to be DNase 1 resistant. Okay, and so half of the capsids that we were making were intact capsids that had all of those properties, and they also had the property that they had the right cesium chloride density, and they had approximately the correct size on a sucrose gradient. Although they were much more heterodispersed on a sucrose gradient than I would have liked to see. The other half of them clearly were DNA sensitive, had approximately the same density on a sucrose, on a cesium gradient. So they had about the same amount of DNA attached to what would appear to be roughly an intact capsid. And they were also infectious, and they were also infectious with a much larger piece of DNA. They could package 2x size DNA. So clearly it was not being packaged. It was just attached somehow to the capsid. But that was good enough to get it inside of a cell and get it expressed. So I can't remember now what I'm commenting on from Doug, but those are basically the facts. We got enough. I mean, there are other things. The correct particles were A20 sensitive, B1 insensitive. The incorrect particles were B1 sensitive, A20 insensitive. The antibody profiles were correct. All of these things were appeared to be correct.
Yeah, the question was though, the source of the capsid protein was cesium purified assembled empty capsids, as best I can remember.
No, we did not. Again, I'm not quite sure what the question is. We did not throw in empty capsids.
I see.
We made an extract that was actively expressing both the replication proteins and actively expressing the capsid proteins.
No.
We essentially used, Doug, your PIM45. So you know that does not have terminal repeats, but it has the internal portions of the genome. And it actively expresses all the reps and all the capsid proteins. That then was turned on with adenovirus, and that's the extract we had. So we had an extract that was actively making in the cytoplasm capsid proteins and rep proteins, but it had nothing to replicate. We then added DNA. We added LAC-Z or GFP DNA that had been isolated as hERT DNA from actively replicating cells. Okay, so that had been purified as DNA substrate. It had TRs. It had a complete genome. A complete marker genome, and that was added to a crude extract that had capsid and rep. So in principle, we may well have been translating capsid genomes in that extract as we were packaging. In fact, thinking about it now, the fact that the capsid proteins are so unstable almost meant that we had to have been translating simultaneously with packaging, simultaneously with replication. So we were replicating, we were making capsid, and we were packaging. All of these things had to have been happening at the same time. I can't imagine how else it could have happened. We could not just throw in denatured capsids. Jürgen tried that experiment, and we knew that wasn't going to work. We thought about throwing in empty capsids. Jürgen tried that experiment. We knew that wasn't going to work. So we just threw it in there. It was a Hail Mary pass. And we were totally surprised that we actually got something. Now, the only thing we got was about 10 to the 7 particles per milliliter of reaction volume, which is not exactly going to change gene therapy. But, we have, presumably there are nuclear proteins that we didn't have that we need to add back. I mean, there are clearly cellular proteins that are involved. About all you can say about that work is that we developed an assay to now use to fractionate cell extracts to find out what cellular proteins are involved and to try to come back together with a total package. And we now know what replication proteins to add. So we pretty much solved that problem. The last part of it was the MCM complex. And unfortunately, although we know we need the MCM complex, we don't know what portion of it we need, and we don't know what accessory proteins to MCM we need in order to make the replication assay work. There's the MCM, which were a complex which was affinity purified, and then when we ended that replication study, was not pure. It was predominantly MCM, but it had CDTs, it had a little bit of gin proteins, all these proteins that are involved in MCM assembly. So we say we know what replication proteins we need to add. We've solved that problem, but we haven't really completely solved it. And there may be some wrinkles in there that still have to be solved. I'm talking now in terms of if we decide to go and get serious about an in vitro packaging, we probably know what replication proteins to add. I'm not sure how we're going to generate the capsid proteins in sufficient quantity before they get degraded in our reaction. Maybe they're not degraded in our reaction. These are all things that we need to answer in order to, as a next step. Anyway, that's enough.
So I want to clarify something because I think there was a little bit of a dogma out there that empty capsids were formed, rep would bind to it and thread DNA. And what I'm hearing you guys say is that that's not the case. It's probably formulating around the DNA or using DNA as a scaffold, and then you're packaging the particle with the DNA almost simultaneously. There isn't a bucket of empties and a bucket of replication, and then the two start interfacing with one another. And then the other thing I'm hearing is that capsids are very unstable, that if you don't have this happen, they fall apart. They don't seem to stay together at any significant level. Although we can make empty capsids independent of replication. So something.
We spent a lot of time. One of the reasons the mutant paper, the capsid mutant paper, hasn't been published because it was really designed to find intermediates. And so we tried tagging capsid proteins in a variety of ways and hope to find the kinds of intermediates that Jürgen saw in his sucrose gradients. And we were never, ever able to isolate intermediates, no matter how we tag capsids. And I don't think that we tried for quite a long time. I don't know whether we just were not very good at it, or what it says to me is that capsids, the intermediates are very unstable. They get destroyed much faster than we can isolate them.
So, guys, Weidong, you wanted to ask a question?
Oh, sorry. I just want to kind of like add something to Nick's observation. And we also did an experiment, extract empty particles and extract the DNA and run the gel. And we also see this 200 base pair fragment. And also did the sequencing, single molecule sequencing. And then most of them are kind of like really just like a AAV dimer. So just kind of two ITRs linked together. And then the other things in my single molecule sequencing of AAV genomes, and I also see a lot of like genomic DNA. It's really random sequence, and always in the kind of like also like in the a kind of like a complementary or orientation fragment, things like that. So I think that's kind of patterning, and I don't know what how they're getting there, but we don't seem to get those sequence in the gel. But I think just like two things observations. That's one is like in the empty particles, in the recombinant AAV production system, those 200 base pair fragments are just AAV ITR dimer, and we also see a lot of like genomic DNA contamination in AAV encapsulation.
Interesting. So I'm going to ask Ken to launch us in a direction and go around, because I know we're time-wise. I appreciate everybody joining. There was a 2-day meeting by the FDA on insertional mutagenesis. Doug actually chaired the session, and there were a lot of points brought up about AAV jumping into the human genome. And I think the numbers that started coming out was, irrespective of what your transgene is, you're getting about a billion integration events happening in the liver at the doses that I was describing to you earlier, and that these integration events appear to be random. They appear to be, illegitimate. They're not jumping anywhere specific in front of promoters or things like that. And yet at the same time, if you think about treating young kids and or repeat dosing, you're putting in a lot of potential and saturation integration events that take place And I don't know of any way to prevent this from happening. But at the same time, I think this is going to be a plague of AAV for here to eternity, which is an uncontrolled recombination event that appears to be, if you do any kind of CRISPR where you go in to edit the genome, you find AAV's already jumped into the site. And if you make these exposed ends or cutting, you get the AAV molecules are in there almost before anything else happens, and they're all so wherever there may be breaks in the chromosome, AAV is a tremendous substrate for jumping into those breaks. I think, Ken, if you could add what your thoughts were about that, and Doug, you chaired the session, if you could give us a little bit of where is the field going, because at the Miami meeting that Arun and I were at, they were actually saying, can we make AAV integrate somewhere specific so that we can control this integration and not be a victim of it? And I guess you guys know that when Unicure had the tumor show up in the hemophilia patient and they take it out and screen it, it did not have an AAV integration that was causative. And there were 2 dogs that were hemophilia 8 dogs at Penn that actually had an amplification of hepatocytes that were expressing factor 8. So the levels of factor 8 went up. The AAV was in those cells, but it didn't look like AAV caused those cells to do anything. It was like a clonal expansion that took place. But now there's a tremendous amount of effort just looking at like saturation bombing, where are all the integration events taking place? And they seem to be, if you look at chromosome pictures, they're all over the place. There's nowhere specific. And yet this is a fairly risky thing to assume nothing's going to happen over time. Or if you think about hepatitis, where you have an insult 30 years later turn into a cancer, we may be doomed at some level by not being able to either manage or be a byproduct of this random integration. And I thought, Ken, if you and then Doug could start this, maybe we could get some take-home lessons about what are our options here.
You want me to start, or?
Yes, please.
Yeah, so, the first thing is, it turns out that if you look at the ITR and its ability to fold up, it's a holiday structure, which is an intermediate in recombination. And so the possibility for AAV to integrate into another DNA is extremely high. I think also, and as far as I can tell, if you put any piece of DNA into a mammalian cell, there's going to be recombination between that piece of DNA and the genome. And the question is, how much and where does it go? I think that with AAV, there's a couple of things that are interesting. One is, depending on how you screen for integration by AAV that you put in, you get AAV all over the place. If, on the other hand, you try and clone out cells that will express transgene, then you start seeing a lot more specificity, and it's going into the chromosome 19 site to a high frequency. So there's a real difference as to what gets in. In some places, it's a small molecule, and in other places, it's the whole genome plus. And I think that that became clear in the French experiments, where they looked at what AAV sequences were present in their, their hepatoma samples from patients. And it turned out that there was a small sequence from AAV from the right end, which included part of the ITR and part of the unique sequence, about half and half. And there was a couple hundred nucleotides that were preferentially in there. So there's something about that right end sequence in particular that seems to promote this sort of thing. Now, the beauty is, of course, if you make if you make your vectors appropriately, you can get rid of the right end, and so that's not an issue, depending on what you're using. Just get two left ends. But I think that I think you're always going to have this this issue. And what's interesting is if the cells are replicating, you get one kind of result. If the cells are stationary, more or less, you get another result. And so the situation is incredibly complex. But I don't think that when you see what I call random integration of AAV, it's unclear to me how much of the genome is getting in. Most of the screens look for the ends of AAV, and that's what's getting in. It's that right end, preferentially, in my opinion, that's getting in. And so that's easy. But if you look for something in the middle of the genome, you get a different kind of result. And if you look for a genome that's functional, you really get a very different kind of result. And so I think that you've got two problems. One is if you're worried about the transgene in your vector inserting in the wrong place, that's one issue. If you're worried about some AAV sequence integrating in the wrong place, that's a much higher likelihood, although it's going to be on a completely random kind of basis. But clearly it's going to go into open areas of the genome where transcription is probably taking place because they're available. And I think that, so there is some targeting. What was interesting when people looked at the hepatoma results was that there was a preferential integration of this short sequence into regulatory regions of genes that tended to be preferentially expressed in these hepatomas, and whether that was a function of being in the hepatoma or not. But in about half the hepatomas, the integrated AAV sequences were outside the region of the tumor. So the whole thing is a big, let's say it's complex.
Doug, I think one point Ian Alexander and others identified that right-hand unique sequence has like a liver enhancer element.
Well, that was very different,
yeah.
But I think what was interesting was in 1983 or 2, when I was with Ernest Winokur at the Weitzman, we looked for an experiment for recombination between AAV and SV40 in monkey cells. And guess what? The recombinant sequence from AAV that was in the recombinants was that short piece on the right end. So that we knew this data 40 years ago.
Interesting.
What it means.
Doug, you want to bring us back to some of your studies with the insertion stuff?
Yeah, so just to reiterate with what Ken said, any DNA with a free end is likely to recombine either with itself or with a break in the chromosome. And chromosomal breaks are going to occur most of the time in active regions, either actively replicating or actively dividing. So, apart from that, the integration is random. You see it all over the place. But what's not random is integrations that actually cause tumors. And, these are, well, apart from the hepatoma model that Ken just talked about, this is all from mice and to some extent from the dogs. But what we saw is if we go in with a vector that is comprised of nothing but a promoter and a strong promoter at that, you see lots of tumors, and it's very easy to find where they integrated just by using a PCR from the ends. When we went in with just a more conventional GFP vector, reasonably strong promoter, enhancer, but protein coding and termination, we couldn't find those, even though we had excess tumors in those animals. And they were a tumor-crowned mouse model, but we certainly had much more tumors when we infected with AAV to just a GFP. When technology evolved to the point where you could do target capture along with deep sequencing, we were able to find those. And the reason we couldn't find them before was that they lacked the ends. They were all truncations of AAV, such that the protein coding region and half the intron was gone. And what was left was the promoter and the splice donor. And the absolutely fascinating thing was that all 5 of those that we pulled out were in one specific gene. They were all in intron 2 of the itch gene, which is in itself a E3 ligase that functions as a tumor that essentially targets a tumor suppressor. So overexpressing the ligase downregulated the tumor suppressor. But my point is, even though integration is random, the mechanisms that actually can cause tumors can be very, very specific. So we know that we had many millions of those truncated vectors in our prep. We looked at all the circularized vector and quantitated, well, how much of it has that kind of a conformation where you've just got promoter and a splice donor. And it constituted about 2% of the total, which is a lot. And so that would have integrated randomly all over the place. But the only place it caused tumors was within that itch gene. Other vectors, such as the one that just had the CBA promoter in it, caused tumors and associated with a lot of different genes, mostly HRAS, FGF2, all the usual culprits. But this was very specific for the itch. Not that it didn't integrate everywhere else, but those interactions that can cause tumors can be very specific indeed. So that I think was the most compelling outcome of that study, which has not yet been published, even though I've tried on several occasions to get it published. It meets a lot of resistance.
So is this a random, I'm sorry, is this a liver problem? Is this, most of the tumors being investigated as liver tumors, or are there other kinds of tumors that have been seen?
These were all liver tumors. I don't know of any other tumor in a mouse that's ever been associated with AAV.
Nothing but liver tumors. And I asked Weidong if he could throw a figure up, because Doug describes an intermediate being generated that's getting packaged, and it's showing up about 2% of the product of the final virus. And Weidong has described recently a mechanism by which these things seem to be showing up as snapback molecules, and Ken might be able to add to this as the old Barry Carter DI particles. But Weidong, can you just quickly help everybody understand the experiments you did and the products you're seeing? Because for me, unless we purge these out of the AAV vectors going into the brains of these kids or into their muscles or livers, they're generating exactly what I think Doug has characterized as the culprits for creating the insertional problems. Weidong, can you put it on full screen?
Yeah, can you, okay, can you see my screen now?
Yeah, but can you go to full screen? There you go.
Can you see me?
Yep.
Okay, so what you see here is like like when we get AAV vectors, we all know that sometimes you get a smear just like beyond the major band, And so I did this kind of like single molecule sequencing, found use a way to identify all the molecules in the AAV population. So that's kind of like a snapback genomes, and we can see, and we also see those genomes with deletions, and we also see those incomplete AAV genomes. So that's kind of like just like the incomplete packaging. And let me see. Yeah, so all sorts of like genomes, but a lot of this like snapback genome is a major interesting stuff, and it's kind of like it's basically like a self-complementary vectors. So, and the mechanism for generating this, and I thought it's from a kind of like NHEJ process, and I thought like that in the AAV production process, and it will generate a kind of like recombinant AAV fragment pool, and this fragment will likely to each other form this kind of like molecules that can like replicate in the package. It's kind of like become subgenomic particles. So like by packaging of these since you will have these symmetric snapback genomes, asymmetric snapback genomes, and also the genome with deletions, and also the other stuff. So I think the key is like I think is like how the mechanism for generating this kind of like a subgenomic population is because of the lesion and the nicking and all those procedures happen during in the AAV genomes. Yeah. And I think like we also I think this is like somehow like it didn't get right. It's let me see. I think the animation somehow get I'm getting I actually get the animation here, so I think like I didn't see. Let me see anyway.
So I think while he's working on that, one of the take-homes is this thing is generating as a byproduct of replication, it appears to be.
Okay, so I think like this is a one second.
And Ken's gonna ask.
So I think like it's for the snapback genomes, I think like there is one like implication is like after this subgenomic particles like snapback genomes, getting like a second generation DNA synthesis, and it will form this kind of dimer, and this dimer will transcript as kind of double-strand RNA, and the double-strand RNA potentially can lead to RNA interference that can reduce the gene expression. That's just one aspect. And another aspect is like, basically is the molecule, Doug has been described in his experiment. And because in some of these molecules, look like this is going to be basically, it's a molecule that's just with only promoter. And for example, you look at here, this is a molecule it's basically a molecule that with only promoter, and just the kind of molecule described in Doug's paper, and it's kind of potentially can be caused like a promoter read-through and so on. And actually, I don't have all the data here, but it pretty much, in the population, we can see this at least, from 0.1% to 5% of this kind of snapback genome existing in the AAV population. So I think this is going to be a significant amount of those kind of molecule, but I think the consequence of such molecule is really unknown, and because it's various amount of different preferable for AAV preparation. And this is the paper recently described by Beverly Davison, and she shows that in the AAV toxicity, and they see this kind of plasmid backbone translation and transcription, and in the single molecule sequencing, and we did show that in a kind of bad situation when the AAV genome getting packaged, and in some of the molecule actually will have this ITR and the promoter and the link directly to the plasmid backbone sequence, and that could potentially explaining why some of these molecules can get transcription of the the plasmid backbone, and it also can explain why this kind of transcription only happen in 3' ITR, and it's not because the 3' ITR has a specific promoter activity. Other than that, because this 3' ITR is actually follow the promoter, and this promoter sequence, connect to the plasmid backbone, and that's lead to the transcription of this kind of sequence. Yeah, I think that's probably all I have.
So before Ken comments, I want to just summarize Weidong's data again. there's a replication mechanism that seems to generate a snapback molecule that's getting packaged. And when he takes the smear on the gel and pulls them out and does single genome sequencing, he's characterizing all of these products being generated. And then they end up looking like the ones that Doug described are the culprits for jumping in front of the itch gene and the intron 2 and promoting expression of that ubiquitin ligase. But more importantly, these products are being generated in the production system, are being packaged. And I think it was Arun, if I remember correctly, that kept showing me that when he overexpressed rep protein, you got more types of recombinants than if you modulated the rep protein, that it seems to bind to pseudocytes or things like that, and maybe nicking. But Ken, I know you want to comment directly on that observation. Then Arun, maybe help us out.
Well, I think that Peter Ward did a lot of replication experiments, and the more interesting ones were that depending on the conditions and whether or not you had you were using an extract from a helper positive cell or one that lacked helper, in the absence of helper, you saw the growing strand fall off the template, fold back on itself, and simply replicate back to the end, and you wound up with a hairpin molecule with a loop on one end. Now, what was interesting was, to me, was that how frequently this happened depend on the length of the genome. If the genome was short enough, like about 20%, you deleted the middle 80% of the genome and just had the two ends and did the replication under those conditions, then you got full-length molecules and you got net synthesis. So it just kept on going, and what it seemed like was that AAV replication, the growing strands hangs onto the template until it gets to the ITR at the other end. Then it sort of gets recharged and starts all over again. And the finite possibility of its falling off during this process, but if the distance to be traveled is short enough, then it's not very unlikely to fall off. So there's a lot of stuff that I don't think we yet understand, despite Nick's good work, about AAV DNA replication in its totality. This is a real example of that. So anyway.
Yeah.
Let me add one quick note. So we did 2D gels at one point, looking at replication in vitro and in vivo, because we wanted to convince ourselves that what we were looking at in vitro really reflected what was happening in vivo. And the interesting thing that came out of that was that you did not get nicking at the site that you had just finished until you got all the way over to the other end. And the only way that could happen is if the new end was communicating with the old end and essentially saying, I'm not going to do anything until we're finished going through this entire strand and displacing this entire strand, okay? Now, the only way you could do that is if rep bound to both ends were interacting with each other. And we know that TRs bind dimers or trimers, but that in fact, when you do a gel chip experiment, what you see are two TRs bound with about a hexamer of rep, okay? And it's three reps to this TR, 3 reps to this TR, and those that are bound to each other. So it's entirely possible, as you were suggesting, that the two ends are talking to each other while you're displacing the strand and not doing something that would cut it in the middle, let's say. And that process could fail multiple times to generate these kinds of molecules.
Arun.
What's interesting is that if you look at Barry's original data, and everybody else's subsequently, was that the number of interfering particles sort of cascaded.
Yeah.
The initial passages, when you had a good adenovirus to AAV ratio, you didn't really see that. But if you kept on passing it, you just build up a tremendous number of these interfering particles.
Yeah, that's true with SV40 as well. I mean, that's something that the interfering particles don't have as far to go to replicate, so they finish faster. They do more. That's my theory anyway.
And they suck up the the reagents that are needed.
Yeah. Arun.
Yes, Jude. So I'd like to make two very quick points. This may be of interest to this general discussion. First of all, I know we pay a lot of attention to the French study that was published by Nault and colleagues in 2016, I believe. And that created a lot of discussion, although there's been quite a bit of pushback. But then since then, there are 3 other papers that come out, one from Korean group, showing that the incidence of liver cancer is very, very rare in their patients. And then 2 more papers have come out, one from NIH, and the more recent one is from Guangping Gao. He looked at 413 patients with 9 different cancer types, including hepatocellular carcinoma. And he sees no difference in terms of the AAV DNA positivity. So he basically has ruled out that AAV plays any role in hepatocellular carcinoma induction. And Jude, you mentioned this. You and I were at this Miami meeting where Marcus Gompe presented some very, exciting data showing using in humanized mice where up to 90% of the hepatocytes are of human origin. When he delivers AAV to these, he begins to see tumor formation. But very interestingly, all the tumors are of mouse origin. He has never seen tumors coming out of human hepatocytes. So I would really be very, skeptical of all the mouse data that have been published. And the mouse are basically, you look at in the wrong way and they will develop liver tumors. So I put very little stock in that.
Well, I think that's one thing that came out of all the discussion with ASTPT and FDA, is that the mouse is essentially a very sensitive model for tumor formation. And that appears to be, related to the level of telomerase, the differences between mice and humans.
And it's also very curious.
That said, sensitivity in a model is a good way to understand mechanisms, and that's what I see the value of the studies. And then on top of that, I think what we have always lacked in the field of AAV is any understanding of the natural history of AAV in humans. We don't know what a viremia looks like. We don't know how much virus the liver ever sees. So we don't know how to put that in perspective with a dose of 10 to the 15th or 10 to the 16th IV. And I think that would be a hugely important thing to understand.
So Doug, one thing
sorry, points one taken, but can I just add one other comment that, in exception to the mouse models, if you look at, rabbits, look at dogs, look at monkeys, and 200, as of yesterday, I looked it up, there have been 272 phase 1, 2, 3 clinical trials in humans. Not one reported case of tumor formation in humans. So again, I think this points to the fact that, mouse models are not predictive of what would happen in humans.
So I think one thing that Marcus mentioned at the Miami meeting was that in the monkey, he was using retroviruses to actively promote the first oncogenic event in the liver, followed by AAV, and he was not able to initiate tumors in those animals. And then he was using the humanized mouse model and trying to precipitate the same tumors that I showed you in that image of the humanized mouse, and he wasn't able to do it. Only in the mouse did he see it. But I think it doesn't remove the risk that Ken brought up, and that is any piece of DNA is going to integrate randomly, and at some point, if you do a high enough number of patients, you're going to see some kind of event take place that may be, you know, an SAE. To me, it's more of how many of these can you accumulate over time before they end up being a factor that influences something else 20 years from now or 30 years from now as part of a cascade of things. But I did want to share with you guys, and I apologize to Dirk and Matt and Chengwen, who actually demonstrated the double-stranded RNA data, not having an opportunity to speak directly, but I did want to share this with you because I thought this was interesting. This is from Ben, who was in Xiao's lab and now is working at the Expo. He was actually looking at the rep and comparing it to CRISPR and trying to model it in a way of determining what were the functional things that were similar to CRISPR technology that are in the rep protein itself and how it interfaces with the DNA and so forth. And he brought up the obvious things. It has an endonuclease activity, DNA binding, and the helicase, exactly the same as what the rep protein was doing. But then he actually showed me this, that the D sequence is basically like the PAM element that you see in the CRISPR, and that the nicking site in CRISPR is an NGG. And I think you heard Ken mention this morning that the heterogeneity in AAV is TTG as a nicking or TG or G itself. And so some of his thoughts were that, you know, this D sequence may actually have serves a purpose of being the equivalent to the CRISPR element of nicking at a specific site or interfacing and creating the nick. And we always had this conversation no matter where we were, where did AAV come from, where did it evolve from, and things of that nature. And I think you begin to see conservation of these things across phylums where the proteins have conserved very specific activities. But I thought just this exercise of looking at what is embedded in the so-called editing proteins and what's embedded in the AAV rep protein, in my opinion, was attractive in the sense that they do look like they have evolved a very similar scaffolding modeling type approach to carry out their activities. The question is, could you take this, and I know Nikki would argue it has to form the hairpin and it has to have the sequence in the right location and everything. Could you begin to use this as a way of directing rep integration? When Ken and team showed it went into the AAV S1 site, that's because downstream of that sequence was the putative AAV nicking site. But could one begin to evolve so-called nicking-like elements and use rep to direct sequences to targeted locations? Just something to think about there as we all go down our holiday.
Has that work been published?
No.
Oh, okay. I'd love to see that when it's ready. I wanted to ask Doug one question real quick. So what was the sense, what were there any conclusions or any sense of what direction the FDA wants to take about this integration stuff?
They have not been very communicative about it. I think they're still formulating their position. But one thing they are doing is requiring, more preclinical assessment of the vector in terms of integration patterns. And we have tried to direct them away from assays based on just observing integration patterns randomly in cells, because we expect that they're always going to be random, or random to the extent that you can measure, and try to push them in a direction where you're actually measuring transformation at some level and quantifying it, and then determining, well, what was the mechanism of this of these transformation events, and do they relate to humans?
Yeah. It ended up being very clear that you can score integration by sequencing the entire single cell sequencing. Integrations appear to be random. The byproducts, I think what Weidong and Doug have been talking about, are probably real, and they are contributing to this. If there was a way to purge those out of your production systems, that would give you a better product. Like Ken said, intact genomes integrate differently than DI particles and other things that are formed in our production systems. And at the moment, the mouse has not been a predictor of what's been seen in the primates or in higher organisms. But as I mentioned to you, the transductions not work the same as what you get in the mouse. The AAV8, which was the gold standard for going into the liver, has failed miserably in both the monkeys and humans. And if it's a capsid transcriptional interaction phenomenon, the field needs to refocus not on libraries, but maybe on what are these interactions and how do they govern whether a genome is functional or not functional. And Jürgen, I was going to give you the one of the last words when we were in the bar with Dirk, and Dirk, I needed to thank you again for getting us together in Heidelberg. You mentioned to me the POP protein, and that you thought that was a very interesting interaction with capsids that people never followed up on or you weren't able to publish. And you're on mute. Yeah.
Yeah. That's something which we had found at the end of, before I retired, of my time in, we did a 2-hybrid screen with the N-terminus of the VP1. And we found quite a number of very interesting interactions. And this screening has been done by 2 independent groups, so not ours, but also a service group, and they come up with the same hits. And among these hits, is the N-terminus reacts with cell cycle components, cyclin A, pCNA, with a transcription factor, TOX4, and a ubiquitin detection system, a protein which attaches to substrates and leads to ubiquitination of the substrate. And this is the SPOP protein. And and this prevents degradation, for example, of incoming new capsid proteins. So you can very nicely show that. But another very interesting interaction is also interaction with Nexin-6. Nexin is a component of the retrograde transport mechanism. And it has been shown that parts of the retrograde system are required for infection with AAV. And it is not clear whether the AAVR or the capsids interact with it, but it seems that the capsid with externalized VP1 N-terminus is able to interact with the retrograde, the retro transport system. So this is Nexin. Yeah, but I don't want to summarize more, but there are definitively hints that the capsid proteins are able to recruit in cis in the infection process, interactions with cellular components which are important for the transduction. And one more point I want to say is that deletion of the VP1 N-terminus has an influence on the trafficking of the endosomes inside the cell. So there is more activity of the AAV N-terminus than we have thought so far. Thank you.
Ken, go ahead, and then Dirk, I'll let you finish up.
All right. I'm on the FDA committee at the moment, and I've been involved in some of these discussions. I think what's becoming more and more a serious question, and we just had a meeting last week, and that is what are the limits of the value of the observations made in the mouse with respect to its implications for humans? At what point can you carry over, or what point, as you pointed out earlier, Jude, with the AAV8, it totally doesn't carry over. And I think this is a serious issue, which has been commented on with regard to the oncogenicity question, et cetera. And so the real challenge for AAV, it seems to be, is to find a better animal model that can afford to use, which will actually to give us more pertinent information in terms of people.
Yeah, good point. Dirk?
I just wanted to very quickly pick up on what you just said, Jürgen, and also what Nick discussed before, the overarching topic that there's more to the capsid. We didn't get a chance to talk about this today, but you just mentioned libraries, Jude, and I think there's a lot of open questions regarding the library screens that we do, and a lot of take-home messages from you, things to think about. If, and that's, I guess, the emerging picture, the capsid does much more than simply deliver the DNA and acts as a transcription factor, and we think about how we screen libraries, be it focused libraries where we take 100 capsids and barcode them, or millions of capsids, and we dump them on cells, and all these different, or many of these different capsids get into the cell and then do more than deliver their own genome. They might insist transactivate other genomes, and then if we screen on the RNA level, this might be a total mess. So I think there's a lot of things we don't understand. We have to decipher biology to translate into screening technology.
And a really good point. And I think it was brought up by Luke at the Miami meeting that we haven't really done directed evolution because our libraries are not complex enough to pull out a capsid like COVID Delta, which has 13 mutations in order to change, that we don't have the ability to do that type of selection. What we're really doing is panning large collections of capsids for functions. But I think your point's well taken when you barcode it, and then you might barcode for RNA and you get a result that shows a good RNA expressing capsid. What is that capsid? Is it doing other things, the signaling cascades, the transcription and stuff like that? So it's interesting. So guys,
Jude, can I make two very quick points?
Yeah.
One is the, I forgot to mention that Kathy High published the results of her first hemophilia trial and followed these patients for 15 years and saw no evidence of any liver toxicity or hepatocellular carcinoma in these patients. And the final point I will make is that, and I've been saying this for a while, that I think we have to keep this in mind, that AAV evolved as a virus, not as a vector. So we can't really expect it, it doesn't express its own genes very efficiently. So we can't suddenly start expecting AAV to, expressing our therapeutic genes to high levels. And clearly the capsid protein, I'm also very skeptical about all these fancy libraries because they are going to be creating a lot of unknown antigenic epitopes. And so I would try to keep it as simple as possible. Just make the vector less immunogenic and more efficient. And I'll shut up.
Thanks, Arun. Listen, guys, I appreciate this. What I might do, if you're still interested, is maybe pick a topic like what Jürgen or Nick or Kim were describing about specific interaction with a cellular protein and what that may mean and things like that, because we have gotten way ahead of ourselves in that the clinical community is going in with vectors with assumptions that, higher dose is all you need. These higher doses are showing thrombopenia with complement, they're showing liver toxicity with unfortunate adverse events. And then there's other variables that are starting to show up, at least at the Miami meeting when all of the clinicians were there and talking. They said, one of them mentioned how the patients all seem to have problems the next day and they're vomiting. And another clinician said, I see the same thing. And then you realize everybody that was in these clinical trials that were getting large doses of AAV had the same problem. And I think Kim and Mike mentioned that viral load put on the patient is so high that trying to handle this is turned into a, almost like an urgent need to disperse all of this vector. And Barry Berns actually put up a calculation that said based on the size of the particle, when you're at 10 to the 14 particles per kilo, the amount of antigen surface is the size of the left side of your lung, just in surface. So we're at these phenomenal doses that, as Arun said, doesn't match with the virus and its life cycle of being, an infectious agent that goes silent versus one that's in there to turn on genes or be pathogenic, and that we're trying to harness this to do something that we haven't been successful with yet, maybe won't. But at the moment, I can tell you that the phenomena of what happens in mice and how it doesn't translate into monkeys and appears to be a mirror image in humans, both at the transcription level and gene function, seems to be the largest impediment right now. That a new capsid doesn't solve the problem. It essentially waits its turn to get into the clinic to find out that it too gets stuck at this level of something's happening at the higher higher species that you don't see in the rodents. If it's a transcription component or recruitment or whatever, it has diverged when you look at mice and you look at monkeys. And if there's a factor, Jürgen, that is binding to these elements that's facilitating, we need to understand that. And I think there's more and more data that the VP1, VP2 are becoming the critical components that make this work. But I really, really appreciate you guys getting together, because I think this is a community that, I don't know, Ken, how many years? 50, 60 years for you? 40 something for Nick and Jürgen, and the rest of us are trailing right behind you. Was
51 years.
51 years. Phenomenal. And yet at the same time, I think we're very fortunate that we watched the evolution of this virus turn into a vector, and there are patients that are clearly benefiting from it. So it's an amazing transformative capability. I can't end without doing my classic, some type of joke or something. So y'all just bear with me while I let you go for the holidays.
Oh, Nick, you're really going to enjoy this one. Now, I know how hard it is waiting until Christmas morning, but it won't be long before my sleigh is flying over, Florida. Say, since we have a moment, why don't we just double check something? Ah, yes, there you are. Oh my, the naughty list. How did you get there? Must be because, well, not everything stays in Vegas. That's right. I've been keeping an eye on you. I better get back to work. But before I do, let me be the first to say, Merry Christmas. I'll see you real soon. 2 lumps of coal. Classic.
Thank you, Santa.
All right, guys. Listen, y'all have a wonderful holiday. I'm gonna